one of the questions I had a bit ago was about, when we talked about accommodation with the eye, was particularly about the rod phototransduction. And one of the things we talked about with that, and it's important to point out again, is that we have to begin with a stimulus, or a lack of a stimulus. That stimulus is light, and the part that's going to pick up or detect the light is our photopigment. For our rods, what is this? Rhodopsin. Rhodopsin is two different pigments together, or two different chemicals together. Retinine, good job. And scotopsin, all right. They've been studying. Now, if you forgot how they can look, you might want to look on page 33. And we see retinine here in the cis-retinine, or retinol it's sometimes called, and trans states. All right, the cis-retinine is the non-stimulated, and it's the big one. It's what it looks like in the absence of light. The trans-retinine, light stimulated. Very nice. All right, the trans-retinine is the straighter molecule, and as it's stimulated by light, it takes a change in shape. And what else does it do? Disassociates or disconnects from the scotopsin. Very good. All right, so this is our trigger. In light, the rhodopsin is stimulated and changes from the cis-retinine to the trans-retinine, causing it to separate from scotopsin. That's the trigger. Okay, do we want to do light first or dark first? Over here. Okay, in the dark, we have an absence of light, so the rhodopsin stays bound together. Because of that, this causes sodium channels to open, and we have an inflow of sodium, which is going to raise the resting membrane potential, making it more positive. As it becomes more positive, this causes a change in the calcium channels as well. The calcium channels now also open, increasing the positive nature of the cell even further. Oh, okay. All right. Now I don't know how to start again. From the beginning. All right, so we, what?
10 words maximum.
10 words maximum? I gotta pick them? Sodium, calcium, becoming more positive, that's too many, and glutamate.
All right.
Inhibition.
Ooh.
Okay, all right, did I get 10? I'm sorry, let's take this more seriously. Okay, that was fun though. All right, so we have the light causing the rhodopsin to stay, absence of light. Rhodopsin stays bound together, and because of that, we have sodium channels opening up as well as calcium channels opening up. Those two are going to push the cell more positive. As the cell's resting potential becomes more and more positive instead of negative that it should be, this is going to trigger the neurotransmitter glutamate to be released from the photoreceptor, in this case the rod. And as it flows out through the synapse, it hits the bipolar cell and inhibits the bipolar cell from sending a signal. So glutamate is inhibitory. Yes?
In axon terminals, are there multiple neurotransmitters and they're released only with certain things? I mean, do they all start in the axon terminal?
Neurotransmitters typically are released from the terminal ends of axons or the axon. Yeah. In some neurons, there is the ability to release more than one type of neurotransmitter. Most of them, like the motor, sensory, the vision ones that we're going to look at, we're going to keep it very simplistic and just deal with one. But particularly when you get to the brain and look at others, there are some that can be much more complex.
So is it inhibitory because it's dark? It's seeing dark, right?
It's inhibitory because the effects of glutamate cause the bipolar cell to have no action. So it's going to prevent the bipolar cell from sending action potentials.
Why would the bipolar cell send action potentials if it's already dark?
Well, that's it. It's already dark, so we don't want it to.
But would it, if without the glutamate, would you get like that, like when you have a concussion and you get all kinds of signals?
So that's causing secondary impulses. So let's think about this in a slightly different way. When you walk into, let's say you've been in a fairly light environment and you walk into a dark room. At first it's hard for you to see. The reason why it's hard for you to see is because the rhodopsin hasn't built up again. It requires time for rhodopsin to build up for you to establish this dark current, which is the glutamate flow, which inhibits the bipolar cells. And as that's happening, you're able to tell very subtle hues in darkness better. All right? That's required for your night vision and your dim light vision. So the dark current is actually really, really important to that. Yes?
So those subtle hues you can see like in dim light, so how are you getting those if all...
Because in this situation, what we're going to do is there's some little photons of light that are going to break the rhodopsin.
Okay, so only
And
if
they're
there's
going to
small
send us
amounts
to the
of
next step.
you're going to receive.
Yeah. Yeah. So in daylight, all of this gets bleached out. But it's in dim light, and what we're going to look at next is in the dimmer lights. So this is in total darkness. And even in very dim light, you're going to have some rods that are in almost all darkness. And that's how you're going to tell the difference. All right, so that was the picture from the book showing you the dark current. They added a secondary messenger, which I didn't need you to mention. So don't worry about the cyclic GMP there. But they did tell you most of the other stuff. A reminder of the two pigments involved, or the two shapes of the retinine involved. Okay, now we take the same rod, but we have small amounts of light hitting it. And what happens is that the light triggers the retinine to do what? Split from scotopsin. So the retinine form that splits it from scotopsin is transretinine, transol retinine. It's the cis form that stays bound to scotopsin. So because the light comes in and stimulates the retinine, forms it to transretinine, we get that separated from scotopsin, which now is going to cause this reaction. All right, so separation of rhodopsin is going to cause the sodium channels to close. The positive flow into the cell stops. We still are going to have other ion channels open and working, and those, particularly like sodium potassium pumps, are still involved. So because of that, we're going to have a change in the membrane potential. It's not going to continue to become more positive. Instead, it's going to become more negative. It's going to hyperpolarize. Because it's hyperpolarizing, this closes the calcium channels as well. So now we're removing even more positive influx. So it's becoming yet more negative. As the calcium channels also close, the whole membrane is becoming more and more negative inside, and we have a decrease in glutamate release. Glutamate, again, was what inhibited the bipolar cells. So as glutamate is stopped from being released, we don't have inhibition at the bipolar cells anymore. All right, so the lack of glutamate on the bipolar cells allows the bipolar cells to now send impulses. So they are now going to become excited. They will have graded potentials, becoming excited. As they do this, they're going to send an impulse to the ganglion cells. And they tell the ganglion cells, I can see something. So they send this signal, and the ganglion cells transfer this signal back to your brain, and you register that you were seeing light. Again, we are in very, very dim light in this particular case. So it's the ability to stop the flow of glutamate, which is again inhibitory, in the presence of light that allows you to see the light. And again, even very low light like starlight can trigger separation of the rhodopsin. Questions? Yes.
Sorry. So if it's like really dark in your bedroom and you can only like barely see something like on a counter or something, and like as it becomes lighter outside, you can see better and better. It just means that more rods are firing, and that's why you can see better. 'Cause, like,
Well,
you know,
is it becoming lighter outside, meaning like the sun is coming up?
Yeah, so like as you have
That's
more
not
light
more
entering the
rods working. That's actually more cones working.
Okay.
So what happens when you go into a dark room and then get used to it is that basically buildup of rhodopsin. Usually when you wake up in the middle of the night, you notice how you can kind of see a little bit?
Yes.
Why? You've been asleep, you've not been stimulated by light, your rhodopsin's built up. So you can see when you wake up in very dim light, and again, it's not real well, but you can see really in this dim light situation because of that, because you've got built up rhodopsin. It's that change as you get to the more light flooding in. We view things very much more clearly in bright sunlight. And we have a lot of those cones in the fovea centralis, so when we see any light that is going to detect color, we view it as more clear, and we're going to have a lot better picture of it. So it's kind of the difference there. We're transitioning to actual color vision, and we're getting out of the dark vision.
And so is there like a threshold difference in the cones that you can like, so that
Yeah, so
you then see light?
the cones are going to need to get more light than the rods will. So rods have a much lower threshold than the cones do. All right, so this is by no means the only thing on the test. So I guess what I'm asking you right now is what other questions do you have? Because I got one question and I just answered it. And I have a lot more questions for you.
Can you give us a hint on what the other essays are?
I already did. Weren't there a couple that I just talked about? There are actually 3 things I just talked about. The first answer is always your name. You guys are going to hate hearing that by the end of the semester. You're not asking me questions, so I'm just giving you the one I can give you. Yes?
We get our name right?
No. No, I won't give you extra credit for that, but I'll give you the points you earned for it, which is enough of it, the reward, I think. There is some extra credit on the test. I will not tell you what it is on, except that it is something that we talked about during this part of the class. If you guess